Science and Technology Center for Integrated Nucleic Acid and Protein. The title of tonight's seminar is Biotechnology, the Genome Initiative and Medicine in the 21st Century.
There has, can you hear me okay? Testing, one. Is this working? Hello? Is the audio, can we turn the audio up? It's no one. Oh, well, the speaker's not working. Oh, he did it. There is and has been a real revolution in biology in the last 20 years or so, and it's been fueled by a series of technologies that in fact are shown on the first slide. And these are technologies that many of you, I suspect, are familiar with. Is it possible to turn off the front row of lights so we can see the slides? Great. Well, I don't know, people might fall asleep if we have them all on. That's great. Yeah, I think that'll be sufficient. These technologies have not only changed how we do biology, they've led to changes in concept about the fundamental ways we think about biology. And my own view is that in the next 20 years, the kinds of changes that we've seen in the past 20, if anything, will only be accelerated. And indeed, the kinds of technologies that we're talking about relate to instruments that initially can manipulate DNA and proteins, that is, the basic molecules of life, more effectively, both for their analysis and their synthesis. And as well, I think in the future, one of the challenges for biotechnology is going to be to determine how molecules interact with one another in networks, how cells interact with one another in networks, and even how organs interact with one another in networks. So we got two of them? Oh, okay. Perhaps no more revolutionary new tool is going to be developed than the tool provided by the Genome Initiative, namely a complete map and a complete sequence of the 23 pairs of chromosomes that are present in each and every one of your cells. And in the course of this lecture, I'd like to show you how we're going to approach this problem and why I believe this book of life, if you will, is going to so profoundly alter how we do biology and as well how we carry out medicine. Now, before we get into that, let me give you just an overview of what, in a sense, I think is really the most fundamental problem in all of modern biology, and that is developmental biology. We all start as a single cell, a fertilized ovum, and we go through a series of events, cell divisions, that lead to spheres and cylinders that flatten and evaginate and then ultimately generate the different organ layers and the 10 to the 14th cells that are present in an adult organism. And of course, there are two critical kinds of questions that are the focus of modern developmental biology. One is the question of how the individual genes are turned on in the right cells at the right time at the right level. So what are the nature of the molecular controls that regulate the temporal and the spatial and the magnitude of gene expression? Those are questions that we really can't even begin to answer at this point in time. And of course, related to that is what I call the choreography of chromosomes. What are the nature of the controls that tie together in networks batteries of genes so that in a muscle cell you express the 5,000 gene products that uniquely give the muscle cell its particular characteristics, and in a brain cell perhaps 10,000 different kinds of genes that give that particular cell its characteristics. It's these fundamental questions of developmental biology that I think I can demonstrate to you that the genome will provide us with absolutely unique opportunities for approaching. Now let me give you a very brief review, since I know the audience is quite heterogeneous in their knowledge, of essentially what 4 billion years of evolution has done in the terms of storing and transferring information. The chromosomes, as you know, are a 4-letter alphabet that is linear, basically, in its dimensionality. And encoded in our chromosomes are of the order of 100,000 genes for a human organism. Each gene expresses a unit of information in a manner I'll describe in just a moment. Now, the genes then are expressed through an intermediate, in a sense, working computer tape called messenger RNA. Again, it's a 4-letter alphabet. The 4 letters are related to the 4 letters of DNA. And again, it's linear in its dimensionality with regard to information. And of course, the final information transfer process is taking this working computer tape and translating it into a protein in specialized machines that give you then a linear polymer that has 20 different types of subunits, the so-called amino acid subunits. And it's the order in which these subunits are joined together that causes the protein to fold in 3 dimensions to make a molecular machine. And it's these 3-dimensional molecular machines, the proteins, that execute all of the basic functions of life virtually. And of course, the real question in modern biology then is, how do we go from the 1-dimensional information of the chromosomes to the 3-dimensional information of the proteins? And one of the fundamental and unanswered questions is, given the linear order of amino acid subunits in a protein, can we set up predictive measures that will allow us to determine the 3-dimensional structure of the protein when it folds? Now, this process of information transfer is read out, in a sense, from the DNA or from the genes, 3 code letters at a time. Each of these code letters represent a different type of one of the amino acid subunits. So you go from 4-letter alphabet, read off 3 units at a time, to a 20-letter alphabet, which then constitute proteins that fold into these molecular machines. Now, from the point of view of the Genome Initiative, the sequence of our chromosomes, number 1, is going to tell us the identity of all 100,000 or so human genes. Now, this is an exacting computational and experimental problem, a very subtle one with a lot of ramifications, and I won't really have time to discuss it. But once we have these 100,000 genes, we will have the basic units for manipulating fundamental biology, and we'll have, in a sense, 100,000 potential therapeutic reagents for medicine to explore in the future. Now, as I've already intimated, associated with each of these genes are regulatory elements, and once we've sequenced the human genome, we will be able to decipher the regulatory code of the genes and to understand these temporal and spatial and magnitude regulatory events, as well as this choreography, the networking together of different kinds of genes. Now, beyond the genes and the control elements, some have argued everything is junk and why should you sequence it? Yet, if you think about what chromosomes can do, you realize that they are marvelous organelles that must have contained intrinsically within them a great deal of additional other information. For example, each of you has in every one of your cells of the order of 2 meters of DNA. That has to fold into 23 pairs of chromosomes that are much smaller than the dot of a pencil. So that is a process of information compaction that modern engineering can't begin to duplicate. Yet what controls this compaction process are proteins which must recognize structures on the DNA. And not only must they recognize structures on the DNA, in order to express a particular gene at a particular point, you have to unfold, you have to deconvolute these chromosomes and let the genes be then exposed to the enzymes of transcription and translation. So how you compact and decompact this in a gene-specific manner requires recognition sequences that are complementary between proteins and the genes that are present on the chromosomes. And it's these kinds of codes that the genome will help us with, quite obviously. Now, the dimensionality of proteins to make molecular machines is really a mysterious thing. If you look at a protein, you can't a priori have any idea what kind of function this protein might carry out, and yet it's an enzyme that has the ability to cleave a certain kind of carbohydrate moiety in the groove that you see on the upper right-hand side there. And of course, the directions for folding these linear chains into these three-dimensional structures can be exquisite in their specificity. And here's an example of a portion of an enzyme whose substrate binding pocket is shown in green. The substrate, an amino acid chain, is peptide chain is shown in orange. So there has to be a complementarity that permits the substrate to fit into the enzyme pocket, and then one has to juxtapose in 3 dimensions the appropriate amino acid side chains to execute the chemistry that permits the chain to be cleaved. And of course, again, the challenge for the future is going to be to design from first principles the capacity to create proteins with any shape, with shapes of any type of complementarity, and to understand how you can juxtapose chemical amino acid side chains in 3 dimensions to execute any particular chemistry you'd like to wish. This is the world of protein engineering, and that's a world that we'll return to later. Now, what has really revolutionized and made possible the Genome Initiative in first principles is the techniques of recombinant DNA. That is, the techniques whereby we have enzymes that can cut DNA at particular sites, we have enzymes that can join DNA together at other sites, and we have enzymes that can synthesize copies of DNA. So those 3 categories of enzymes, restriction endonucleases, ligases, and polymerases, are the essence of which the revolution of molecular biology has come. And it's that revolution that then has permitted us to seriously consider this incredibly audacious task of mapping and sequencing the entire human genome. Now, to a first approximation, there are really 2 kinds of maps. There is a genetic map wherein we can take human traits and we can follow their segregation through families, and if indeed they are linked together on a single chromosome, the distance of their separation will be proportional to their ability to co-segregate in family studies. So you can generate these so-called genetic or recombinational maps where traits that are physically very close together will appear close together on a genetic map, and traits that are distant will appear distant on the genetic map. Now, we want to be able to translate genetic maps into physical maps because we want to have our hands on the DNA that encodes the gene that causes schizophrenia. And that's where the second category of map comes in, and these are called the physical maps. Whether they are maps that are created by locating the sites at which these DNA-cutting enzymes can cleave double-stranded DNA, a so-called restriction map, or whether it's taking the smallest or the largest manipulable unit of DNA, which today ranges in the order of 1 to 300 kilobases, 300,000 bases of DNA, and to put these fragments together in a linear jigsaw puzzle so that you get an ordered library of these fragments that extends across the entire chromosome that you're interested in doing. And of course, with these pieces of DNA, you have each of them in a test tube, and when you can then link a particular genetic trait to a particular DNA fragment, you have in your hands the gene. Now, in a sense, the ultimate physical map is the sequence, that is, the exact order of these 4 different subunits for every single chromosome. And again, the task of mapping and sequencing the human genome is enormous. These 23 chromosomes contain more than 3 times 10 to the 9th base pairs of these DNA subunits. There are really 24 different chromosomes. The 2 sex chromosomes are distinct from one another. They encode 100,000 genes, and with a process called RNA splicing, the amount of information that one can generate from these genes is totally unimaginable at this point in time, but certainly much greater than the 100,000 genes that you see there. Now, a part of the Human Genome Initiative is the understanding that model organisms should also be analyzed. And there are two categories of model organisms. One is a complex organism like ourselves, the mouse, which will be used both as a model system for human disease and to examine and identify conserved sequences in the chromosomes. And by identifying these conserved sequences, we will be able to locate regions that evolution has said cannot be changed because they have predetermined specified function. And of course, these functions may encode proteins, that is, they may be genes, they may be regulatory elements, or they may encode some of these other functions that are associated uniquely with chromosomes, such as folding and compaction. Now, the second category of model organisms are those simple organisms such as bacteria and yeast to begin with, and then the more complicated worm and fly and so forth, because these simple model organisms are going to be the keys to telling us how human genes work. By relationships of similarity or of homology, we can take a human gene and find in the yeast its counterpart quite often. And in that simple yeast, we can manipulate those genes in ways that are totally impossible in humans to determine, at least in that organism, what its counterpart does. And armed with this knowledge, then we can go back and look at the human genes. Now, the timetable for carrying out the Genome Initiative is, in many of our views, of the order of 15 years. And the first 10 years, from my point of view, should be spent with a major focus on technology development, because we need tools for mapping and for sequencing, and perhaps most important of all, for analyzing all this information that are somewhere between 100 and 1,000 times better than the tools that we have now. During that time, we can do some mapping of chromosomes, and we can do a very modest amount of sequencing. But it is in the third 5-year period that I suspect we'll have very powerful, fully automated tools, extremely powerful informatics systems that will allow the remaining 95 to 99% of the human genome to be sequenced easily within 5 years, and as well many other things. It'll probably be done in a few large centers, I would guess by subcontract. What that will mean is biologists will never again clone or sequence genes, because it will be done and it will be sitting in your computer waiting for you to carry out the analysis. So the basic objectives of the Genome Initiative are, number one, to develop technologies, and this is the first biological initiative that has ever had as a major focus technology development, and in that sense it marks a real milestone. And then number two, it's to give us computer access to the genome sequence and the genome maps so that we as biologists can design the experiments we'll need to do in the future to solve these very complicated network interaction problems. Now, let me talk then about the benefits that are going to come from the Genome Initiative. And they really fall into three categories that I'll talk about: some of the new tools that we'll have, how it's going to change the practice of biology, and how ultimately it's going to change the practice of medicine. And what I'd like to do is spend just a few moments talking about the approach that we've taken at Caltech toward technology development in a few limited areas. About 2 years ago, we received the Science and Technology Center's award for a center in essentially molecular biotechnology, and that permitted us to put together groups of scientists that have virtually all of the expertises that you see here. So computer scientists, engineers that could do instrumentation, people that were expert in protein separation and analysis and in microsequencing, chemists that were experienced in nucleic acid chemistry, and from these have come the specific interests that you see there on your right. So we are focusing on 2D gel electrophoresis as the most powerful separation method, and we're using it as a preparative tool for obtaining protein for sequence analysis. Clearly, the future in protein chemistry and sequence analysis is mass spectrometry, and I'm really not going to have time to talk about those two things tonight. But what I will talk about are the approaches that we've taken toward DNA sequencing, toward setting up facilities for large-scale sequencing, and toward the techniques that we're developing for DNA diagnostics and mapping. And what we'll start with is the whole question of DNA diagnostics and mapping. Now, it's quite clear if you think about it from first principles that one of the fundamental problems in the Genome Initiative is how to create genetic maps. That is, how to create maps that tell us the relative distances of genetic traits that we'd like to examine with respect to one another on chromosomes. The techniques that exist now for the cognoscenti are using restriction enzyme fragment length polymorphisms and using either simple or more complicated kinds of repeats in ways I don't really want to describe. I will just say that the first two approaches toward genetic mapping are in the future not going to be useful because they aren't fully automatable. And what I'm going to tell you is about a technique that we've developed at Caltech that is called polymorphic sequence tag sites that is powerfully automatable, and I'll show you then how it can be applied to the Human Genome Initiative. So the basic idea in this technique is to be able to take advantage of the enormous genetic variation called allelic variation of human DNA sequences. Your chromosomes differ from mine by probably 1 in 500 bases. So each of those differences are potential genetic markers that I could use to map particular regions of your chromosomes. So what we'd like to be able to do is to develop a simple automated test for analyzing these single nucleotide genetic variations. And I described at an earlier seminar today the simple approach that we've taken to doing that, which is to look at a particular target DNA that has a site of variation and to construct two DNA probes, each about 20 subunits in length, that are adjacent to one another, and the blue probe has its end to the right, the 3' end, right at the site of the mutation. And to the blue probe, we put a hook so we can pull it out of the reaction mixture. To the red probe, we put a fluorescent reporter group so we can identify it easily. And then the basic idea of this analysis is to be able to hybridize those two probes to target DNA. When there is a perfect match of complementarity probe to target DNA, one of these classic enzymes of recombinant DNA techniques, DNA ligase, can actually covalently link the two oligonucleotides together. Then you can use the hook to pull out the blue probe, and it pulls with it the red probe, and you see a nice little red spot that says you have molecular complementarity. In contrast, if there is a lack of molecular complementarity, if there's a substitution there, when you pull out the blue probe, you don't bring the red probe, so you get no color. So in a sense, it's a very simple barcode analysis. And indeed, we have taken this analysis and coupled to it a technique called the polymerase chain reaction, which allows us to amplify target sequences we're interested in examining for genetic variation by 1,000,000-fold, carrying out the ligation assay and doing this all in a little well that's present in one of 96 wells in the microtiter plate. And we have a robotic workstation that automatically does all of the chemistry that's necessary basically for these reactions. So we can, in this automatic mode, carry out the analysis for more than 1,000 of these polymorphic sites in a single day. Now, to give you an idea of how effective this is, here is an example of the assay carried out on normal genes and mutant genes using respectively normal probes and mutant probes. And you can see it's all or none. It's either black in this black and white slide, or it's white. And in fact, what we have is a detector sit down on the 96 well rate plate, read off the answers from each well, and then a computer does the calculations that can draw out a genetic map or do the DNA diagnostics in this particular case. So it is an extremely powerful technique that allows us to carry out this kind of analysis. Now, it's quite clear that this analysis is going to replace, at least in a formal sense, this kind of analysis will replace the other approaches that we've talked about for genetic mapping here, because it's a barcode, it's a plus or minus, and in the future we'll be able to print out a barcode identity plate for each human individual that can be used in a variety of ways to uniquely identify you. There isn't any electrophoresis and there's no radioactivity, and it works on small fragments, and it's fully automatable, and we can detect any kind of substitution that occurs. And then there are some other technical things that I won't go into. And finally, it's absolutely compatible with computers, because it's going to be a barcode absolutely analogous to the barcode that you have at the grocery store. So you can see the technique is enormously powerful in many different ways. Now, we're going to use this technique to map the human chromosome, and the chromosome we've chosen for a variety of irrelevant reasons is chromosome 14, and this is a chromosome that has 100 million base pairs in it. And the approach that we're taking then to mapping the chromosome is basically to use a cell sorter to purify chromosome 14 from all other chromosomes, to shear it up into very small pieces, to determine the sequence of 150 or so of these pieces, and then to use that sequence information in ways I'm not going to discuss to identify the sites of these very common polymorphisms, and then to make the automated assays that I've talked about here, the OLA assays that I've talked about here. And then when we're finished, what we'll have is 150 or 200 of these markers that are on chromosome 14, but we won't know their location. The way we'll determine their location is by taking these 200 markers and looking at 40 families that have the parents and children and grandchildren. And essentially by analyzing the markers that we have in the context of these families, we will be able to align all of the markers that we have in the context of those families. And that will create a genetic map that can then be used in the future to ask whether there's any particular disease phenotype that correlates with any particular region on chromosome 14. We've carried out sequence analysis in a preliminary fashion in exactly the way I've described, and the important point about this slide is that of the sequences we look at on chromosome 14, more than half of them exhibit polymorphisms. So the polymorphisms are common, are very easy to get at, and to carry out this kind of analysis that we're talking about here is going to be a straightforward kind of thing. Now, for those of you who have any interest in genetics, I'm going to make a technical point that will be irrelevant to most of you. The informativeness, that is, the useful of two alleles as genetic markers, is limited by the fact there are only two alternative forms and not 4 or 6 or 18. But what we found in a number of sites is a series of genetic markers that are within 400 or 500 base pairs of one another. And it turns out, in the two cases we've examined in detail, they are in partial linkage equilibrium with one another. They make many different haplotypes, and the collection of those 4 markers in one example together have a heterozygosity of greater than 80%, it turns out. So it turns out that you can use nested sets of these markers to give you very informative genetic markers. The other point that I can make is if we generate 150 of these markers, on average, that will give us a marker every 2 million bases, and that will be a good genetic map that can be used as the first screen to locate on chromosome 14 any gene that predisposes one to a particular disease. Now, what is, I think, really exciting about this idea is it takes an earlier idea that we proposed in a paper in Science and makes it infinitely more useful. We suggested that for physical mapping, a really terrific way to join together the large fragments of DNA that will be necessary to cover an entire chromosome would be to take a 250,000 base pair fragment, to sequence either end of it, and then to use this polymerase chain reaction to generate a unique signature for each end of these sites. And that unique signature is called a sequence tag site. And it's nice for all the reasons that I've listed over there. And I'm not going to go into the technical advantages, except to say this approach gives us a language for doing physical mapping, and we can store all of these clones not as a physical clone, but as a sequence in a computer. And that gives one enormous advantages. And what our approach has done is just to take these sequence tag sites and add to them a genetic marker right in the middle of them, and to give them enormously enhanced power. And of course, that enhanced power then gives us the capacity to tie together the physical and the genetic maps. Because with these polymorphic STS sites, we can use the polymerase chain reaction to pick out the large clones that correspond to each of those 150 sites or so. And with those clones then, we've reduced the problem of creating a physical map from a single problem of 100 million bases to 50 problems of 2 megabases, and that's an easily solvable problem. So polymorphic STS sites can join together the physical and the genetic maps. So it's going to be an extremely powerful tool in the future, not only for doing genetic and physical mapping, but for automatically being able to screen through very large populations of human beings to see whether particular phenotypic traits, schizophrenia, correlate with any one of the chromosomes that have been so characterized. Now, let me talk about a second type of analysis, and that's going to the ultimate physical map, DNA sequencing. And of course, the basic approach in DNA sequencing is to insert your DNA into a vector that has just a single strand of DNA, and then use a complementary oligonucleotide that's been radiolabeled at one end to initiate DNA synthesis into the unknown region. And if you add to the mixture a dideoxynucleotide that lacks a critical hydroxyl group for DNA chain extension, you can cause a few percent of the chains to terminate, in this case, at every single A all the way across the unknown sequence. And of course, if you do 3 other reactions for the other DNA letters, you can create an array of fragments that can be resolved by analysis on gels, and then you can use the ladders to translate the DNA sequences. And what we did at Caltech about 5 years ago was to develop a way of automating this technology by color coding each of the 4 different arrays of fragments that terminated at the different bases, by mixing them all together and running them in a single lane of a gel that could resolve nucleotide fragments that differed by a single base. And at the bottom of the gel, you'd activate fluorescence with a laser beam, and then you'd have a photodetector, photomultiplier tube that could essentially read into a computer the 4 different colors, which then could be translated into the DNA sequence at the end of the fragments as they migrate by. So this instrument then has been automated by a company, Applied Biosystems, and the current instrument can actually carry out simultaneously 24 of these analyses and read off the color coding. And in fact, here is a fingerprint of a human gene carried out by this type of analysis. And typically we can get 450 to 500 bases in this analysis. Now, what one can't begin to appreciate with this simple discussion is the complexity of what is going to be necessary for very large-scale DNA sequencing in the future. And in fact, the part we've automated, that is the electrophoresis and the reading out of the sequence, is the easiest part of sequencing now. The really difficult parts are the front end, where you have to prepare the DNA and carry out the sequencing reactions. And the most difficult of all is the back end, the informatics, where you have to put together all the little strings into the big string that is a particular human chromosome. Now, the front end has been automated in terms of preparing DNA and sequencing DNA, using this robotic workstation that I mentioned earlier, the BioMech, and we can carry out all the sequencing reactions that are necessary to feed these machines very, very easily. And in fact, we can use a machine like this to prepare the DNA that we need to carry out the sequencing reactions as well. So what we're now in a position to be able to do is to take DNA from one of these little bacteriophage at the beginning of the day and carry it through these automated procedures and carry out the sequence analysis, in fact, at the end of the day. So it's truly a remarkable feat that's possible at this particular point in time. Our strategy for sequencing these large fragments of DNA is to prepare inserts that are of the order of 35 to 40,000 base pairs in length, and to break them up into really small pieces and to put them into these M13 clones, and then to sequence 600 or 700 of them, and then ask a computer to assemble together the little strings into a bigger string. And with 700 fragments, we can assemble strings that cover 98% of the sequence, and the last 2% is generated by using a chain polymerase reaction, making probes from either end of the contig. So the procedure is a very straightforward one, and the hardest part of it is the computational task of assembling all the little strings into the one big string. And it turns out to be an incredibly difficult problem because there is error in sequencing, and it's computationally very difficult to take that error into account. But to give you an idea of what these sequencing machines can do, we can run 24 samples per day and 450 base pairs per run, and we can thus get more than 10,000 base pairs per day, and at 5 days a week and 52 weeks per year, we can get 2 million base pairs worth of raw DNA sequence, more than 2 million. Now, if that sounds terrific, but there are two constraints on it. One, we can't keep up with the rest of the things to really keep the machines running at that rate. And in fact, for the last year, we've probably used our machines at the rate of 15 to 20% of their actual productivity. And number two, with this shotgun method we've talked about, you have a 6-fold redundancy. You sequence, on average, the same thing 6 times over. And that's terrific to bring down your error rate, but it certainly is wasteful in terms of the actual sequence that you generate. And in addition, we have control sequences in there that we have to sequence. So by the time you're finished, you go from 2,800,000 base pairs down to 300,000 to 400,000 base pairs. Now, we're going to make the machines 50% more effective in their throughput. We're going to cut down the redundancy by a factor of 2. And then this will, within a few years, put us in the position of sequencing easily in a year 1 million base pairs. The future, I think, is very exciting for DNA sequencing. All of these techniques, and I'm not going to discuss them, could lead to breakthroughs where you could think about doing tens of thousands, if not hundreds of thousands of base pairs per day, very easily. The technology that, in my view, in the near future has the most potential for contributing is mass spectrometry, and there are at least 3 or 4 different ways that one could think about using that in the future. Now, I've spoken about the problem of information analysis, and here is an example of a gene that we've analyzed in an interesting way. And the problems of informational analysis relate to the acquisition, the management, the pattern analysis, there's a pattern of a gene, if you will, and its distribution. But the problem with the gene that I've shown you there and its analysis is it's one that is not susceptible to being extended. This is a gene that on its side is only about 5,000 base pairs. Here's what the gene looks like. It's roughly 1/1,000,000th of the human genome. And if you had to amplify this 2-dimensional pattern analysis array by 1,000,000-fold, it would obviously be computationally impossible to handle. And in fact, the major calculations that are necessary for analyzing genes, that is, looking for similarity, turn out to be incredibly computationally intensive. That is, they're exponential in their demand on computers as you increase the size of the database and the yearly input of DNA sequencing. So slide is 2 years old. The database then was 25 million bases. The yearly input was about 10 million bases. To do the most rigorous similarity analysis of those two things against one another would take the largest supercomputer then almost a year to do. So you cannot analyze the information in the genome by simple, even supercomputers that operate in a sequential fashion. You have to carry out parallel types of analyses of one sort or another. And what Tim Hunkapiller has done at Caltech for the last 3 years is develop a series of super chips that convert into silicon particular algorithms that are essential to fundamental pattern analysis in biological problems. So a chip that we've collaborated with TRW in making, the Fast Data Finder chip, is optimized to query large databases for patterns that you can define and specify completely. Whereas a second chip we've just finished in collaboration with a mathematician, Mike Waterman at USC, and two jet propulsion engineers, this BIS chip has been designed to carry out searches of local similarity, that is, to carry out these homology or similarity searches. So in a sense, this chip can search for unknown patterns. And it turns out the chips are incredibly complementary one to another. Both of them operate by virtue of a pipeline architecture, that is, the pattern is ingrained in microprocessors, a series of cells that are placed in series one to another. The entire database is streamed through it as rapidly as you can transfer the information. Each cell analyzes the streaming data with regard to its own particular pattern. When it identifies it, it takes it out of the stream and then passes it on down in parallel to the downstream microprocessors for analysis. And at the end of this time, this incredibly short period of time, this parallel processing approach allows you to sweep through and analyze patterns at an incredible rate. And in fact, to give you some idea of what these super chips are going to do, here is a comparison of a Sun Spark station with regard to cost to efficiency ratios against a Cray 2, against a connection machine. So Cray is a supercomputer that's sequential, a connection machine is a highly parallel computer, and then this BIS chip in a Sun workstation is highly parallel but incredibly inexpensive compared to the two things above it. And you can see the incredible price-to-performance ratio. And our idea is we will design a series of chips that inculcate these fundamental biological algorithms and put them on either simple workstations or even PCs to give then biologists supercomputer capacity for particular kinds of calculations. Now, computationally, another problem is the database problem. In a few years, we'll have 100 different databases, and we have to figure out how to interlink them together so that you as a biologist can traverse through them and get the kind of information that you'd like to obtain from it. It's quite clear then that there are a whole series of problems in biology whose essential answer essential solution must come from computational analysis. So whether it's 1, 2, or 3-dimensional image analysis, string theory, predicting the folding structures of proteins from their linear amino acid sequence, or the database problem, we have a whole series of very powerful tools to point at these problems, and we and others are in the process of pushing this very hard. Now, if you think about it, the real problem we have is there aren't very many students that could live both in the world of biology and computer science. So what we'd like to do at Caltech is actually have the biotechnology problems always driven by leading edge biology to develop these tools, but to create an educational program where we would bring in students in biotechnology that would essentially have a double PhD program. They'd take good hardcore biology, they'd take, in this case, good hardcore computer science, and the students would have two mentors. They, in a sense, would be the media for communication between the biologist and the computer scientist. And of course, the exciting part about this kind of concept is you could have students to do this for all of the other disciplines that surround biology and bring in then from those disciplines very powerful new tools to biology, and at the same time you'd create a series of biologists slash whatever that would, I think, be real leaders in biology in the 21st century. And this is a program we've already started with several students on, and it looks like it's going to be able to achieve its objective in all ways. And in fact, the students that we're really interested in many ways are those that have very strong physical and computational backgrounds. Well, let me move to the biological advantages then for the Genome Initiative. One is it's going to let us identify all these 100,000 different human genes and figure out interesting experiments to decipher how exactly they carry out their functions. And it's important to emphasize then how much that is going to change biology in the future, because in the past we all started with biological assays and went to proteins and used the protein sequence information or antibodies to them to get the genes. In the future, we'll have all the human genes and we'll have to devise completely new tools for figuring out how they carry out their functions. And this is going to be an incredibly important challenge, because there are many genes in humans that we don't have the tools for identifying now, short of going to the Genome Initiative. For example, more than half your genes